Okay. So, kita start balik. So, first soalan, untuk soalan ACA, soalan ni adalah draw a half view of an animal pollinated flower. So, apa yang soalan ni nak? Dia nak kita draw apa? Half view of an animal pollinated flower. So, I think we all know, but okay, key point kat sini nak animal pollinated kan. So macam biasa kita kena ada stigma dengan style untuk female reproduction. And then kita kena ada anther and filament untuk male reproduction. And kita kena ada petal, petal dia punya luarannya kelopak bunga tu. And then maybe kalau nak bagi nampak pollinated sikit, buatlah dalam kurungan coloured ke apa. And then kita ada sepal yang untuk hold the flower. And bawah tu kita ada peduncle. Tapi peduncle ni dia tak penting sangat lah kot. So maybe tak familiar, tak dalam syllabus kita. Tapi the key point is flower yang kita hafal macam biasa je. And then kalau boleh bagi nampak anther tu dia macam filament yang hold anther tu dia macam panjang sikit sebab animal pollinated kan. So macam yang siapa dah ada sebenarnya tu kan bila dia punya filament dan anther panjang lagi senang lah pores yang ada dekat seeds yang ada dekat anther tu untuk melekat kat bulu haiwan yang lalu. So that's the whole key point dia. Okay. So next. Kalau ada soalan tanya je lah tau. Okay, next. Dia kata outline apa? How the hormone auxin controls phototropism in plant shoots. So dia nak tahu macam mana hormone auxin ni control phototropism in plant shoots. Okay, so yang kita ingat phototropism ni apa? Phototropism tu bila plant tu shoots tu elongate towards matahari lah, sun. So the first point kita nak cakap is phototropism berlaku bila plant shoots grow ataupun bend towards the brightest light ataupun sun. Dia pergi kat cahaya matahari kan. So, and then macam mana dia berlaku? Macam mana? Firstly, auxin akan move from the lighter to shadier side of shoot ataupun stem tip ataupun apex. So, macam mana auxin ni move daripada lighter to shadier adalah through the auxin efflux pumps. So, auxin efflux pumps yang akan promote lah auxin move from lighter to shadier. So, apa yang auxin ni buat? Auxin akan promote cell elongation ataupun growth ataupun causes cell wall acidification. Acidification yang akan explain lagi next slide. Okay, and then point E. More growth on shady side of stem due to auxin concentration gradient. Sebab kita tahu auxin ni dia move kepada shadier. So auxin ni akan causes growth dekat bahagian shady tu lah, bukan kat bahagian light tu. Okay, and then point last ni dalam mark scheme. Auxin ni dia akan binds to auxin receptor in target cells. So nanti bila auxin tu dah bind dekat auxin receptors, dia akan promote expression of genes for growth ataupun secretion of H+ ions into the wall. Okay, next slide. So apa yang jadi actually, tadi tu sebab soalan dia 5 markah je. So it's okay kalau you guys outline macam tu je. Tapi ni just untuk revise balik. So apa actually jadi? Auxin ni in shoots, auxin increase the flexibility of the cell wall to promote plant growth via cell elongation. Okay, macam kita tahu tadi, auxin kan dia akan move from lighter to shadier side. So apa jadi actually? Auxin tu dia akan activates the proton pump in the plasma membrane which causes the secretion of H+ ions into the cell wall. So sekarang ni kita actually nak generate proton gradient tau. That's why auxin akan causes secretion of H+ ions into the cell wall. So apa akan jadi lepas berlakunya secretion H+ ions kan? So kalau kita ingat H+ ions ni dia causes something to jadi makin acidic sebab H+ ions kan. So itu yang membuatkan ada decrease in the pH dekat dalam cell wall tu yang causes cellulose fibers within the cell walls to loosen. Ataupun kita boleh cakap by breaking the bonds between them. Maksudnya cell wall tu kan dia jadi kuat sebab antara komponen dia is cellulose fibers kan. So bila berlaku ni secretion of H plus ions, dia jadi makin acidic, pH dia makin rendah sebab H plus ions tu, and so cellulose fibers tu makin longgar, makin loosen sebab bond between them break lah. Okay, and then additionally, auxin upregulates expression of expansins, which similarly increases the elasticity of the cell wall. Expansins ni also is a type of hormone. Hormone ke enzyme lah. Kejap. Kejap. Okay, dia protein, so enzyme lah actually. Okay, so auxin ni dia tolong regulate lah expression expansin yang function dia sama je, iaitu bukan function sama tapi function expansin ni nak increase elasticity of cell wall. So bila makin elastic, makin senang lah plant tu nak grow. Macam tu. So sekarang ni number 4, if the cell wall are now flexible and influx of water yang to be stored in the vacuole causes the cell to increase in size. So sekarang ni bila dah makin concentrated sebab H plus ions, berlakulah influx of water through osmosis. So bila makin banyak water masuk dalam cell wall, yang saya tengok kat gambar bawah tu kan, H plus ions masuk dalam vacuole tu, yang kanan tu, yang vacuole tu, so makin lah increase in size plant shoot tu, so makin berlakunya growth of plant towards the sunlight. Macam tu. Faham ke? Okay ke? Okay, kalau tak ada soalan faham kot. Okay, next slide. Okay, so next question. In hot, dry conditions, plants lose water rapidly due to transpiration. Explain how the structures and processes of the plant allow this water to be replaced. So sekarang ni, key points dia kata plant akan lose water due to transpiration, especially dalam hot and dry condition. So dia suruh kita explain macam mana structure and processes of the plant allow this water to be replaced, so that plant ni macam keep on tak mati lah kan, tak layu lah kan. So obviously kita akan explain more pasal transpiration. Okay, so the first point is kita tahu transpiration tu apa. Transpiration is evaporation of water in leaf ataupun mesophyll, creates tension ataupun low pressure ataupun negative pressure yang akan menyebab berlakunya pooling force ataupun transpiration pool. Meaning kita explain lah macam mana transpiration berlaku. Itu transpiration berlaku bila berlaku evaporation of water kat stomata tu. So nanti tension dalam plant tu berkurang, low pressure. So berlakulah pooling force daripada stem tu pergi leaf, iaitu transpiration pool. Okay, and then point B, water drawn through cell walls ataupun out of xylem by capillary ataupun action ataupun adhesion to cellulose. So macam mana actually water ni move? Water move through the cell walls melalui adhesion. Iaitu macam kita tahu adhesion ni kan attraction between water dengan dinding cell wall tu. Kalau cohesion dia water tu water punya bond. Tapi kalau adhesion water through the cell wall. Okay. So, C, bila berlaku evaporation, so kita tahu water leave the leaves through the stomata, so berlakulah low pressure ataupun pulling force in the xylem sebab water keluar, so pressure dalam tu makin kurang lah kan. So, and then kita explain juga pasal mechanism, pasal structure sebab dia nak sorry. Dia kan nak pasal structure dengan proses kan. So kita cakaplah pasal structure of the water. So D tu, hydrogen bonds make water cohesive. Ataupun kita cakap hydrogen bonds ni allow water to be pulled up under tension, atau kita cakap lah allow the transpiration to. So tadi kita explain pasal adhesion, which is the bond between water dengan cell wall. So sekarang ni kita explain pula pasal cohesion, iaitu hydrogen bonds between water and water yang membuatkan water tu boleh flow lah, naik. Macam tu. So sekarang ni kita dah explain proses dia kan. Taklah the whole process, tapi kita dah nampak dah macam mana isi transpiration berlaku. So next ni kita explain pasal structure dia pula. So structure je, kita tahu kita pakai xylem kan sebab nak transport water. So xylem ni dia resist tension ataupun low pressure ataupun dia tak collapse. Macam mana? Sebab dia adalah thickened dengan lignified walls. Meaning ataupun kita cakap lah lignified walls ni prevent the xylem from collapse ke. Macam dia maintain lah structure tu supaya tak collapse. And then kita tahu transpiration ni berlaku bila water travels from the roots to the leaves in xylem. Sebab transpiration pool kan. So daripada roots ke leaves lah in the xylem, through xylem. So water punya absorption. So and then kita explain lagi water absorbed in roots by osmosis. Meaning macam kita tahu lah mechanism transport of water is pakai osmosis from high water potential to low water potential. Okay, and then H, tadi kita explain pasal water. So sekarang ni kita nak tambah lagi, explain lagi. Sebab dia suruh explain kan tadi, kita explain lagi pasal macam mana active transport of ions, iaitu solids into the roots enabling osmosis. So macam kita faham kan bila berlaku active transport of solids into the roots, meaning concentration ions of ataupun solid dalam roots tu dia makin tinggi. That's why bila makin tinggi, meaning water potential makin rendah. So water potential yang tinggi daripada luar roots tu bolehlah diffuse masuk dalam roots. And then also structure lagi. Structure roots tu dia ada deep ataupun kita kata wide ranging extensive root systems ataupun kita cakap many roots hair yang dia increasekan lagi surface area untuk water absorption. Sebab soalan tadi kalau korang ingat dia kan kata in hot and dry condition kan. So at the center kita kena explain lah apa structure plants yang actually hidup dalam climate hot and dry ni. Okay, and then J, ada juga structure je juga ada thick ataupun waxy cuticle yang reduce transpiration ataupun water loss. Yang ni dekat leaf lah, macam yang kita ingat kan, dekat leaf kan ada waxy cuticle. So apa function waxy cuticle? Wax tu dia prevent macam excess loss of water, of transpiration lah. Okay, and then K, plant yang hidup in hot and dry condition, dia juga ada small ataupun no leaves ataupun reduce surface area of leaf, leaf yang kecil lah, ataupun thorns instead of leaves untuk prevent too much water loss lah, macam cactus semua tu. And then I, structure lagi, plants yang hidup in hot and dry condition ni dia ada few stomata. Ataupun stomata tu dia ada dalam pits ataupun root leaves, meaning dia macam makin hidden from the sun, so makin susah lah nak berlaku water loss. So dia boleh preserve water. Okay, and then dia juga ada hairs on leaf surface to reduce airflow near the leaf ataupun reflect sunlight. Meaning dia ada daun yang berbulu lah. So bulu yang dekat daun tu dia macam macam kalau angin lalu kan, dia tak adalah water tu terus tebang-tebang. Tak tahu korang faham kan? Macam bulu-bulu kat daun tu dia macam increase barrier lah untuk angin. So tak adalah berlaku banyak sangat water tebang-tebang, berlaku excess loss of water. Okay, and then last sekali, and stomata open at night ataupun kita explain pasal chem physiology to reduce water loss. Ni actually ada kaitan dengan guttation. Kalau korang ingat guttation tu dia air embun lah. Macam pagi-pagi actually dia still berlaku water transport through xylem tapi in very slow process lah malam-malam. Itu yang guttation tu. Itulah. Okay, ada soalan tak? Kalau tak ada, itu je daripada saya. Sekian, terima kasih.
Okay, move on. So next question, plants have widespread influences from food chains to climate change. So for question A, draw a labeled diagram of the internal structure of a seed. Or B, explain the process of water uptake and transport by plants. And C, describe the process of bed formation. So since C ni tak ada kena-mengena dengan plant biology, kita takkan discuss. Kita discuss A dengan B je. Okay, kita tengok A dulu. So let's draw a level diagram of the internal structure of a seed. So ni internal structure of a seed, and then boleh tengok dekat sini. First kita kena ada plumule. So plumule ni the embryonic shoot, dia yang kita tengok sini, it appears as a bud, dia yang akan give rise to the shoot nanti. Lepas tu kita kena ada radical juga. Radical ni yang akan develop into the primary root of the plant nanti. Lepas tu kita ada micropyle. Micropyle ni dia allow water to enter the embryo before active germination. Okay, lepas tu kita kena ada cotyledon. Cotyledon ni dia akan provide a source of food. Lepas tu last sekali kita kena ada testa or seed coat. Dia the outer coat of the seed yang protect the embryonic plant. So lukis je semua ni label, dapat 3 markah. Oh, seed coat ni jangan lupa kena lukis ada 2 line macam ni. Okay. Kalau ada apa-apa stopkan . Okay, next. For question B, explain the process of water uptake and transport by plants. Okay, so question case study, structure and process. Kan? Kalau ini process of water uptake and transport. So soalan ni tak minta structure. Okay, so first kita nak explain the water uptake dia and transport sekali. Root hair, dia absorb water by osmosis. Lepas tu kalau kita mention osmosis, actually kita dah dapat 2 markah yang berlainan. So root hair absorb water, 1 markah. Water is absorbed by osmosis, 1 markah. So dah dapat 2 markah kat sini. Lepas tu kita kena mention yang solid concentration inside the root is higher than in the soil. Ataupun water potential inside the root is lower than in the soil. And this is due to the active transport of ions or minerals into the root. Okay, so yang ni transport by plants. Okay, so next kita kena mention yang transport of water ni happens in xylem vessels and the flow of water is from the roots to the leaves. Okay, lepas tu kita kena mention juga water movement in xylem is due to pulling force or transpiration pull or such or negative pressure potential. Macam mana water uptake ni boleh jadi dekat xylem, and this is due to the pulling force or transpiration pull and semua ni lah. Lepas tu ini transport okay, cohesion ataupun macam kita cakap tadi, the hydrogen bonds between water molecule tu, cohesion ni yang allow water to be pulled up in xylem. And then next point, transpiration in leaves ni dia causes apa? So transpiration in leaves dia generates tension or pulling force or suction. And then next point, evaporation of water from cell walls. Oh, yang ni transport water evaporate from cell walls. And then addition of water to cell wall, they create tension. Addition ni hydrogen bond between water and the cell wall. So addition of water to cell wall, they create tension forces. Lepas tu L ni actually the structure dia, lignin in xylem walls or thickened xylem walls, they prevent collapse or resist tension. And then last kali kita mention je, movement of water in xylem is a passive process. So dah dapat the process of water uptake and transport by plants. Ada question ke? Kalau tak ada, next lah.
Sekejap je. Alright, so for this question, A, draw a labeled plant diagram to show the distribution of tissues in the leaf of a dicotyledonous plant. So, be careful that for this question, diminta distribution of tissues in the leaf. So, maksudnya jangan lukis jangan lukis like the one yang stem dengan root. Because this one dia minta leaf. So because dia minta distribution of tissues in the leaf juga, so you have to draw the all the cells yang ada in the leaf tu, macam palisade mesophiles, spongy mesophiles and all that. Okay, next. Okay, so this is the kind of diagram yang you have to draw. Ada dia punya bahagian upper epidermis tu, kena ada upper epidermis tu, which ada dia punya palisade mesophiles. And then ada dia punya spongy mesophiles and air spaces kat bawah tu. And then draw the vascular bundle, the one yang in circle tu. Yeah. Draw the vascular bundle, make sure the xylem and the phloem is clear. And then yeah, dekat bawah tu, make sure you also draw also the bahagian lower epidermis, including the stomata. So bila dia minta distribution of tissues of leaf, okay, remember because it's leaf, so kena ada palisade mesophiles, spongy mesophiles, the vascular bundle and the stomata. The 4 things I think are very, very important. Okay. I guess that's it for drawing part. The question balik. Okay. And then outline the metabolic processes that occur in starchy seeds during germination. So dia minta outline the metabolic processes in starchy seeds during germination. So we know that when like a seed to germinates, they also respire. So bila a living being is respiring, it means that glucose is needed. So we just, I think like at least in my head, macam I ingat because dia nak germinate and dia nak respire. So you have to like outline basically how a seed tu, from a seed dia boleh like produce glucose for respiration. Okay, that's the basic idea I think. Okay, so kalau kita tengok the mark scheme, apa tu? Firstly, kejap. Andi cuba next. So kalau we like refer to this diagram, then the first step in germination of starchy seeds is water must be absorbed to activate cells. Okay, water must be absorbed to activate the cells, and then once the cells are activated, baru the synthesis of gibberellins can happen. So synthesis of gibberellins is actually
important for the synthesis of amylase. So water activates cells, which leads to synthesis of gibberellins, and gibberellins would pula akan like in turn synthesis amylase. So the function of amylase is to hydrolyze the stored starch in the seed tu jadi maltose. Okay, so that's the point of having amylase. So to hydrolyze stored starch to maltose, and then from maltose yang dia dah hydrolyze tadi tu, dia akan be absorbed by plumulin radical. Tapi this one I tak sure lah. You compulsory tak tu, right? Okay, but after dia dah the amylase have like hydrolyzed stored starch to maltose tu, further hydrolysis akan break maltose into glucose, which is used for respiration. So this is how yang I cakap tadi tu. When you remember seed germination, you need to remember yang they respire, which requires glucose. So macam mana the seed tu akan produce glucose? You have to remember the steps lah, from like how water activate the cells, and then after the activate the cells, activate the metabolic. So dia activate the metabolic processes, and then gibberellins is produced, and then after gibberellins is produced, amylase is produced, which is used to hydrolyze stored starch to maltose, and then it went for further hydrolysis to break maltose into glucose, and then that glucose is used for respiration. Okay, let's go back to the mark scheme. Okay, so kalau dekat sini kata outline, ada 6 marks. So we can see macam water is absorbed by the seed, or seed is rehydrated. Water is absorbed by the seed to activate the metabolism. And then after water, gibberellin. So gibberellin is synthesized, and then it stimulates the production of amylase. Like we discussed already, gibberellin stimulates the production of amylase, and then that amylase digests and hydrolyzes starch to maltose. And then maltose, akan like further hydrolyze to glucose, which will be used in aerobic respiration. And then that glucose used in aerobic respiration tadi tu is also used for the production of cellulose. So basically, like, just remember water. Water activates, and then gibberellin synthesize, and then gibberellin synthesize to simulate production of amylase. And like amylase, dia hydrolyze maltose to hydrolyze starch to maltose, and then hydrolyze maltose to glucose, and then glucose used for respiration. Okay, that's basically it lah. Okay, for question C. Okay, thank you. So explain the like independent processes of photosynthesis in plants. Okay, this one is in chapter 8 kan, tapi macam I guess you guys don't mind kot buat this question. Okay, like independent tadi. Okay, next. Lagi. Okay, so kejap kejap. Okay, so like independent reaction. So like independent reaction ni yang happens during night time kan. So I guess like the most important part kalau nak jawab this one, like kalau nak explain processes, is like to remember macam the big points. So macam kalau like independent reaction, you have to remember the three key phase lah. The 3 key phase dulu, and then baru macam you explain like the details from the 3 key phase tu. So like the 3 key phase untuk like independent reaction is carbon fixation, reduction, and regeneration of RUBP. Kalau you tengok like in the cycle tu dekat carboxylation of birubisco, that's the first phase, carbon fixation, and then reduction, that's the second phase, and then regeneration of RUBP. Okay. So, but I'll just explain the processes and nanti kita tengok the mark scheme. Okay. So for carbon fixation, for carbon fixation, the Kelvin cycle ni dia akan begins with a 5 carbon compound of RUBP, and then because carbon fixation happens, it means that the Rubisco ni helps the end of our sentence. RUBP ni the Rubisco, which is the enzyme too, actually helps to catalyze the attachment of carbon from carbon dioxide to the RUBP. Okay, that's the first step. So the carbon fixation can so they fix carbon from carbon dioxide to RUBP by Rubisco. So now RUBP to from like 5 carbon jadi 6 carbon. But because like 6 carbon compound is still unstable, so they can break down into 3 carbon compounds, which is glycerate 3-phosphate or GP. Okay, and then so bila dia dah break down jadi glycerate 3-phosphate, okay, now we will go on to the second phase, which is reduction. So from that glycerate 3-phosphate tadi tu, 2 molecules of GP tadi tu, dia akan convert it pula to triose phosphate, to 2 molecules of triose phosphate. So glycerate 3-phosphate and triose phosphate, dia ada the same number of carbons lah, ada 3 carbons kan. But how dia converted from glycerate 3-phosphate to triose phosphate is through reduction. Okay, reduction here, dia refer to glycerate 3-phosphate tu, meaning glycerate 3-phosphate is reduced by an ADPH, meaning glycerate 3-phosphate tu yang gain hydrogen. Okay, so glycerate 3-phosphate tu dia reduced by an ADPH, so an ADPH convert from an ADPH jadi NADP plus. Okay, so NADPH and and then like it also like requires ATP lah to provide energy. So NADPH and ATP ni, these are the chemicals yang was taken from like dependent reaction tadi tu. Okay, like dependent reaction. Okay, and then after the being reduced, there is the third step, which is the regeneration of RUBP. So tadi so bila dia glycerate 3-phosphate tu dah reduced by NADPH, kan dia dah jadi triose phosphate. So this triose phosphate pula only like one triose phosphate yang dia akan pakai untuk jadikan glucose. So for each cycle, only like one triose phosphate yang akan pakai untuk generate untuk like produce glucose lah. So 2 cycles are needed untuk dapatkan 1 glucose. Okay, so what happened to the triose phosphate yang tak pakai tadi tu untuk buat glucose? Okay, that triose phosphate akan go back jadi RUBP. So that's the 3 steps: carbon fixation, reduction, and regeneration. Okay, so now we'll go to the mark scheme. So sebab dia suruh explainkan the process of like independent reaction. So okay, but before you start explaining the processes too, you can tell like okay where this like independent reaction take place. So it take place in the stroma of the chloroplast, in the stroma. Okay, and you can also say juga yang macam you can also say juga after you say yang apa tu, the Kelvin cycle to occurs in the stroma, that it requires that energy, that there are like chemical energy are provided from the like dependent reactions. You letaklah kat mana-mana, kalau you nak letak dekat bahagian reduction tadi pun okay je, but yeah, this is the first 2 points lah. So you can tell the first 2 points and you can tell before you start explaining the process of like independent reaction. Okay, so occurs in stroma and a chemical energy provided by like dependent reaction. Okay, and then Okay, kan like I said, kalau macam kalau macam kalau I tulis essay, I would macam I can like list down the big processes first, like the big phases dulu. So macam like carbon fixation, reduction, and regeneration of RUBP kan. So okay, we go to carbon fixation first, and we like baru explain the details of carbon fixation. So macam this one, the first step is carbon fixation kan. So the explanation to carbon fixation is carbon dioxide is fixed to RUBP by Rubisco. So when you say carbon dioxide is fixed to RUBP, you'll get 1 mark. Tapi when you say carbon dioxide fixed to RUBP by Rubisco, you get 2 marks. 1 point for carbon fixation punya point, basically carbon dioxide being fixed to RUBP, and another one for the enzyme. Okay. And then you teruskan je. So what happens? What happens after the carbon dioxide is fixed to RUBP? Dia akan jadi 6 carbon compound, and then that 6 carbon compound, because it's unstable, dia akan break down jadi glycerate through phosphate. So bila dia dah break down jadi glycerate through phosphate ni, waktu ni baru you can start cakap about like the reduction process tadi tu. So glycerate through phosphate will be reduced to triose phosphate by an ADPH. Okay. I think that one is only 1 mark to say that glycerate through phosphate be converted to triose phosphate by reduction using an ADPH. Sorry, that's 2 marks there. Glycerate through phosphate converted to triose phosphate by reduction is 1 mark. Using an ADPH, that would give you like another mark. So that's 2 marks there. And then so bila dia dah reduce, dia akan jadi triose phosphate kan. So and then explain that because despite kita dah masuk bahagian yang regeneration of RUBP kan, so cakap yang triose phosphate tu is used or like is converted to form glucose. Sebab tadi kan kita, sorry?
Nak tanya.
Yeah.
Yang reduction tu, actually describe yang NADPH jadi NADP ke the part yang phosphate jadi triose?
Yeah. Dia describe glycerate through phosphate tu jadi triose phosphate. Sebab dia gain hydrogen kan, so yang gain hydrogen tu is glycerate through phosphate. So yeah.
We can see macam glycerate phosphate is reduced to triose phosphate ke? Okay, thank you.
Okay. 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 Oh, yang tu dah cakap kan. And then like most 5/6 of triose phosphate used for regeneration of RUBP tu sebab I think tadi because I tak ingat lagi. Okay, tak apa. Tapi for the next point tu, you can say like pasal like only one triose phosphate is used for glucose. So like the other molecules of triose phosphate yang are not used for glucose production tu will be used for regeneration of RUBP. And then the last point, ATP used to regenerate RUBP. Kejap. Oh, and then the last point ni, I rasa dia macam it's just a general point to bagi tahu the function of ATP sebab tadi kan kita dah explain pasal like triose phosphate used for the regeneration of RUBP. So the regeneration of RUBP tu requires ATP. So that's a point. Or you can also macam cakap yang ATP ni is also used to convert glycerate through phosphate to triose phosphate. Tapi this one, yang ATP convert glycerate through phosphate to triose phosphate tu, kalau you wanna macam write the acid tu, bila you nak structure tu, yang tu you can put dekat bahagian bahagian dekat reduction tu. Okay. Sekejap, nak cakap apa ni? Yeah, so I think macam I think kalau you remember the full explanation to like independent reaction tu macam dah boleh dapat like 2 marks lah for each processes tu, macam like carbon fixation, reduction and regeneration of REBP. Okay. So I think that's all. That's all. Ada any questions ke?
Dah habis.
Hello. Arham, Fakrul.